In sheep brain dissection lab 2, we will make 4 coronal sections of the left half of the brain, identify and draw the structure visible at each level. First coronal cut will be at the level of the basal ganglia, which is this level here. We'll make very narrow cut. Second coronal cut will be the level of the thalamus. Third coronal cut will be at the level of the rostral or caudal part of the hippocampus. And the fourth cut will be on the more caudal part of the cerebral hemisphere, where we will try to cut at the level of the superior colliculus and midbrain. First coronal cut will be at the level of the basal ganglia. Basal ganglia are part of the deep gray matter, and they are important for initiation of movements. On this cut, we will observe gray matter, white matter, corpus callosum, this is lateral ventricle, and then we will identify some of the basal ganglia. This is caudate nucleus or nucleus caudatus. This is internal capsule, which is white matter. This is putamen, and this is globus pallidus. Second coronal cut is at the level of the thalamus. Let's again identify major parts, which you can see here. This is gray matter, white matter again, corpus callosum, lateral ventricle. So this big structure here in the midline is thalamus. So this is the internal capsule. So this is again thalamus. Third coronal section will be the level of the hippocampus. So this is very caudal part of the hippocampus here. So let's review some structure which we saw in previous sections. Gray matter, white matter. So this is lateral ventricle still. And so here is hippocampus. And again, this is caudal part of the hippocampus. Fourth cut will be on the most caudal part of the cerebral hemisphere. So this is cerebral hemisphere here. And this structure here is midbrain. Actually, this is more caudal part of the midbrain. This hole here is cerebral aqueduct. This region contains structure called substantia nigra that is important for initiation of the movement and also belongs to the basal ganglia. So in summary, in lab 2 we will cut coronal section of the brain at the level of the basal ganglia, thalamus, hippocampus and midbrain. This concludes lab 2 sheep brain dissection demonstration.